So welcome everybody to this series. The theme of this one, very much close to our hearts, is the quantum mitochondria, energy, information, and implications for health and disease. And today it's a pleasure to welcome, and I'll introduce them more fully in a little while, James Moon, Professor James Moon, and our own Professor Alistair Nunn. And thank you very much to the other speakers that have agreed to speak during this program. We're particularly pleased with the program, and it looks quite exciting this time round. Welcome to all participants, and also welcome to those who are new to the foundation. Now, we've been convening these online symposiums, I said, since March 2020. I'm reading from Nina's script here, so I get it right, from March 2020. And we've explored topics ranging from the fundamental concepts of quantum mechanics to their application in new therapeutics, and that is a giant leap. Now, a consistent theme has been the importance of mitochondria, and with this in mind, mitochondria will be the focus of this series. We're exploring how mitochondria respond to different conditions, how they have optimized their function with respect to the Earth's specific magnetic and gravitational fields, and we'll be considering the implications for health and disease. Now, the opening session today is titled COVID-19, Mitochondria and the Quantum Underground, and I'll introduce the speakers in a moment. But first of all, can I just do a little bit of housekeeping? As you know, we record these lectures so that they can be put onto our website, and we now have our own YouTube channel, and a lot easier to pick those up on the YouTube channel. We ask that you keep your questions to the end so that we don't interrupt the lecture. But if there's something, if you completely lost the thread or you want to interject, please do. We can always probably edit it as we go along. But we don't record the Q&A session, so one can feel very free to have, as we have done in the past, very in-depth and interesting Q&A. So without further ado, I'm going to introduce both of our speakers so that one could just follow on from the other seamlessly, as long as their slides are ready. So James is Professor of Cardiology and leads the cardiac MRI at Barts Hospital. Barts is St. Bartholomew's Hospital, and that, to be more precise, to use its name, it's the Medical College of the Royal and Ancient Hospital of St. Bartholomew, and the hospital next year has its 900th anniversary. It was founded in 1123. Now, James is the winner of the 2022 Society of CMR, that's the MRI and cardiac medicine. He won the gold medal, and he has a research index of 93 with 40,000 citations from his 450 papers. So we need to listen very closely to what he says. And he focuses on the better understanding of rare and common heart conditions by creating new tests and linking them to treatment and delivering them to global care. Now, during COVID, he founded the COVID Consortium, and in his words, it was the world's best study of mild COVID, and he's going to be telling us about that in his lecture. And now his latest venture is he's founded Myocardium AI to deliver superhuman explicable AI to the NHS and global care. So James, welcome to the Guy Foundation, welcome to the Autumn Symposium, and we'd be very happy to receive your lecture now. Thank you.
Thank you for inviting me. I am a cardiologist, and I do feel a bit like a fish out of water in such a diverse, multidisciplinary meeting. I have to say that one of the most impressive things I've seen in recent years is some of the animations on YouTube of how the electron transport train works in mitochondria, which really wasn't understood when I was at university. So when I saw these things, I found it incredible. But I am an imager, and my voxel size is 1 millimeter by 1 millimeter by 1 millimeter. So to me, I see 50,000 myocytes at a time, let alone organelles, and let alone proteins, and let alone actual hydrogen ions. So you think in an era and a domain that I aspire to, because nature weaves its threads, you know, a la Feynman. But usually I'm thinking about imaging. So I thank you to the Guy Foundation, and I think I'll explain later how you funded us and what happened, because it's quite a story in itself. So I trained lots of people around the world in cardiac MRI because it was a new technique in diffusing. And I was watching what was happening. And I have a colleague in Bergamo in Italy, which is the sort of first place in the Western world, non-Chinese world, to fall over. And I was thinking that my esoteric cardiac MRI wouldn't be particularly useful. But I had a team that was very tuned in to doing research, applying for grants, getting permissions. And I really thought that you wouldn't be able to ventilate your way out of this. And so the question was, what could we contribute when we couldn't do cardiac MRI? And I went through a number of options. And actually, we ended up thinking about this in a sort of strategic way. And it was my estimate that from the first locally transmitted case to the inability to get research off the ground would be 14 days. Given the 14 days, everything else follows. So you would have, if you were going to do research, you had to do one topic, deeply resourced, ideally at a site with no casualty, which I was at, keeping it simple and maybe empowering others in a new or aspirational collaborative way. And if you're going to do it, you did it all in, or you didn't do it at all. And we published that eventually in a sort of simulation on what would happen. And it was pretty accurate as to what would happen, but actually we got the recovery time. We were over optimistic on recovery time. It took a long time to get research back to working. So what I decided to do was take our hospital staff, who would always come in, and take samples from them weekly, every day from before the pandemic hit to afterwards, and then we'll see where we go. And provide those to basic science groups to do discovery with whatever techniques they had. So I set that up, and the idea was on Friday the 13th. I had finished a proposal and got my colleagues sort of assemble a team on the Sunday evening. I submitted for ethics on Monday. We had the ethical approval on Tuesday at 10 o'clock, which actually made me cry at the time. It was quite extraordinary. We then had 100 documents to write because of the bureaucracy in the site. And the way we did that was every time somebody said, I'm happy with you doing this project, but you need to ask X, I'd just make a new team. So the usual sort of explosion of bureaucracy, we used a sort of explosion of staff to do this. And by the 20th, 9 days or so later, or 7 days later, we had all our permissions. And then 9 days later, we had recruited 400 subjects, taking swabs and bloods. We couldn't get anything analyzed, so we just took them at this time and then assembled teams afterwards to use them. It was an interesting time. We used various models of how to deliver a project. And this was a sort of military model where you think in terms of stuff, staff, systems, and space. And I just alluded to systems we had to set up, and staff we had to beg, borrow, and steal everywhere. This was very good for morale, actually, because everyone says how hard healthcare workers worked during the pandemic, but actually the main thing that struck me was how enormously disruptive and how allocated resource was. So you had, I at one stage was an ITU nurse or assistant nurse, I'm not good enough to be a nurse, and they are amazing people, for a couple of weeks in both the first and second wave, just helping roll patients and all of that sort of thing. It's not an efficient use of your resources, but it was what was necessary at the time. And when we did this, we couldn't use any stuff that might be useful for the NHS. So we had to find what we needed from around the world. And we needed people who were going to use the samples. And this was a group of virologists. Now, at the time, virologists are the lowest in the pecking order of basic science, because virology was done. There's a bit of HIV, but not interesting. In fact, their high grade lab was decommissioned, a CL3 lab, and they had to get it painted over the weekend. So you think about this as all high tech. Actually, it was getting a lab recommissioned in 36 hours on a weekend. And here they are dealing with not yet COVID samples, but they will. They a bit more PPI for COVID samples. We joined a bunch of different universities together. I think we had about 12 or so universities working on our samples, and it's quite industrial. These are the sort of samples for PBMC isolations that this team was doing. This is Marla Maney at UCL. I think we isolated about 100 litres of PBMCs in the end. The total sort of sample size we ended up is in the international units for large study samples, which is in the bathtubs. And I think we had about 3 bathtubs of blood by the end of this project. We did our 400 healthcare workers within 9 days, and here they are. Note there's no mask mandate yet. And because we were managing, we pushed it to 500, and then we pushed it to the Royal Free as well, and then we pushed it to Nottingham and merged with them. And so we had a total of 1,900 subjects, which allowed us to really access genetic aspects of susceptibility. But what was really vital in this was those first 400 samples where we got the initial bloods before the pandemic hit. And we're the only people in the world to have done that. We did this with no funding. We just decided to start and sort the funding out afterwards. And we had to do that all the time. We had to say, we will do this, and find and trust that it would happen. And it did. And you can imagine it might be the Wellcome or the British Heart Foundation or these organizations that could pivot and fund quickly. But actually, it was the pointy end of capitalism, Goldman Sachs, Citadel hedge funds, and a few key foundations and pharma companies that really funded us. So I started with my mortgage, I tipped 40K into just getting it off the ground. And then we crowdfunded from just like a Just Giving website, got 400K off that. And the rest came from these sort of very atypical sources, as you can see there, quite an unusual model. Later, we were part of 8 grants, but because we'd been funded, we just enabled other people and tried to give as many samples as possible so that people had appropriate controls. One of the things that people kept doing was describing phenotypes as severe disease, but they didn't know what mild disease was. And our healthcare workers had mild disease. So what happened? What results did we get? The first thing we showed that no one else showed was that that we took swabs right from day one weekly, and those swabs, when they were eventually analyzed, and it took 10 weeks or so to analyze them because there was no PCR, showed that 7% of our staff in hospital in that first week were walking around with COVID without even having typical symptoms, and that there was an epidemiological curve as it fell. Lockdown was incredibly powerful. So if you hear, that some group got the big calls right, that lockdown was incredibly powerful, but if it had been 3 days earlier, it would have halved the deaths in the first wave. So here is the epidemiology that showed that. It also showed that there was likelihood of intra-hospital transmission, and it was one of the things that prioritized vaccinations and PPE for healthcare workers. Important paper got into the Lancet. We then showed, I started linking up with Public Health England, and we started analyzing at scale the samples, and we started to see, we were the first to describe immunological waning of antibodies, which is a perfectly normal response. It's not right to have, your immune system working at 1,000 miles an hour, and it's supposed to rise and fall, and then recrudesce when needed. So we were the first to show that. We then started working with Imperial. Virology at Barts, immunology at UCL and Barts and Porton Down, and then the T-cell stuff at Imperial. And we started to link the immune system responses to antibodies and T-cells, as you can see here. The graphs are duplicated, but one's in order of T-cells and one's in order of antibodies, showing how some people were very different to others. Now, these papers, I'm putting this funny sort of circular torus on, and these are the altmetrics, and they're an indication of how much things were tweeted or in the media. And we started to get quite a lot of press at this stage. So, many of our papers, some of the papers would have a few hundred press articles, for example. Overall, this study was about 1,200 news articles. We looked at severe disease and started seeing that the heart was damaged in severe disease. and it seemed to be complicated patterns. It wasn't the myocarditis story that everyone was saying. There seemed to be infarcts, and of course infarcts are probably a thrombotic event, and we were seeing lots of pulmonary emboli, and we were the first to show, actually we weren't the first to show that it was infarcts. The Chinese were the first to show, but everyone ignored it because the human tendency is to make things simple, and sometimes you overdo it. Actually, there was quite a lot of ways that the heart could be injured in this early study. You'll remember the scare from a German group saying that lots of people, 73% of people, were having myocardial inflammation. They were using the techniques that were the techniques I had developed, and they didn't do them with adequate controls. In fact, we think they didn't have any controls at all. And so I scanned our healthcare workers, because of the 400 people we had at Barts, actually total of 153 got COVID whilst we were watching, if you like. And of those 153, one person went to hospital for about 4 hours. So they really were mild patients in these, quite ethnically diverse, but all working, average age 40 healthcare workers, 2/3 of them female. So we looked ourselves to try and combat the sort of media stuff at what we found. And what we found was really that the people who'd had COVID versus the people who hadn't, at a few months later, 6 months later, you couldn't tell any difference in their hearts. What you did find was quite a lot of abnormalities. So if you measure a lot of bits of someone's hearts, if you're alive till you're 40 onwards, things are starting to get a bit frayed at the edges. And without adequate controls, you can misinterpret that as causal for whatever process you're investigating. All the basics of science are needed if you want to do science in a hurry. And just because there's a pandemic does not give you the excuse to do your science in a sloppy manner. Otherwise, you will mislead yourself and society. There are lots of ongoing studies now. So we did those, but then the sort of platforms 9 months later came in from the conventional funding sources, and I'm helping run quite a lot of these and do the analyses. The next thing you might have noticed was the vaccine came through, and the thing we saw first was that if you'd had prior infection and then you got the vaccine, it acted like a prime boost. And you didn't really need the second dose at this stage. And that was quite significant. We were in a publication race, and it felt like every day mattered at this stage, and we managed to beat Nature and New England by 2 weeks getting this into the Lancet. And there was a full focus and process on how to get something published that fast. We took the samples, we got the samples by Friday afternoon, got them to Porton Down on Friday 5 p.m, analyzed them by 9 p.m, saw the results were exciting, reanalyzed them with the latest tests, and freezer dived for the old samples on Sunday. Paper written by Monday, submitted Tuesday, published a week later. It was a real buzz. And this immediately influenced national policies. So France and Spain de-emphasized second doses to prioritize them as first doses for those people who'd had an infection in the past. So it made a big difference. First science paper came out when we started looking at T and B cell responses after first vaccine. Another sort of quite high media story as we were starting to look at all the different aspects of the immune system here with really just Wuhan. Later, we're going to get into variants of concern. And it was quite nice to see this. And that was just, Professor Fauci presenting our paper in the White House, which is not something that's ever going to happen to me again. I don't think cardiac MRI really gets into media presentations at the White House, but so properly influential research in a timely fashion. One of the things we saw was because we'd taken the samples before the wave came through, we had this precious 10 mils of PBMCs. 10 mils of peripheral blood mononucleosides is a tiny amount, but to spin up a team to sample and isolate those 80 samples a night for 9 days, The 2 people who did it were working till 3 in the morning every day, incredible commitment from them. And then we put them in the freezer waiting for what the use case would be. And the use case was this. And what we found was that some of our highly exposed healthcare workers never got infected. And when we looked at their serology, before they were exposed, they looked like they had already been exposed. And this was cross-reaction with common cold viruses. In fact, a particular bit of the virus, not the spike, which varies a lot, but the RTC, the replication transcription complex, which is much more preserved across a sort of family of coronaviruses. And that sounds compelling, but the real clincher was that Maddie, my colleague, who's an infectious disease doctor, had insisted on taking a PAX gene tube for RNA. And what you found in those was that the RNA in the peripheral blood for interferon, so an innate immune protein, was innate immune cytokine, was higher in the people who had this sort of exposed seronegative thing. And these people never became PCR positive. And what they had done was clear the infection before the virus had even started to build its spike, because it has to transcribe the earlier steps first. And so that was published in Nature. And it was great to see how the basic scientists build up their story. And this was very influential. This RTC is highly conserved and has now been used. This paper has been used to design a pan-coronavirus vaccine, which was done in Cambridge, and I'm not quite sure what's happened to it, but I believe it was going through into sort of phase 1 studies. This is more about how the red lines are the conservation of this coronavirus genome, and you can see that red is highly conserved in the proper clockwork, which has not got perhaps so much freedom to vary as the spike protein and things that face the immune system proper. This got into the top 0.1% of Nature papers, and it's my only Nature paper ever, so I'm quite pleased. And then later, another science paper starting to look at the variants as they emerged. So we kept changing the platform. And by this stage, we were really narrowing into a group of people who had been serially bled, as they had Wuhan, Alpha, Delta, and later Omicron. And by this stage, you were realizing that serology wasn't going to work as a marker of protection, because, you know, an antibody to Delta wasn't necessarily going to be very good to Omicron. And you started realizing that we had an exposome, if you like, and that we were not all the same. You can imagine just how complicated a population is as you see this wave of infections and evolutionary arms race going through. And then the last sort of or most recent science paper was post-Omicron. And we started to talk about sort of immune imprinting and the boosting you got if you had prior infection. Well, this paper started to show that if you'd had some prior infections, you were more vulnerable to Omicron. It was so than if you hadn't, and Omicron was looking very slippery. A lot of this is quite focused on spike and doesn't fully account for all the T-cell variation, but that's still ongoing work. During this, we had to invent a few methods as well. This is a neat one. We got the spike variants, stuck them on a plate, and pulled down antibody in people's bloods, and were able to measure the relative proportions of antibodies binding each of the different spikes, which is like a neutralization experiment equivalent. And then, of course, antibodies also pull down complements, so we were able to see those differences and to isotype the antibodies. And this is a high throughput platform, which we patented and published. It's nice because it's a platform here, it's using spike, but it could be using any antigenic substance, particularly usually proteins, obviously, but not necessarily, and allows you to really interrogate what's happening to the humoral immune system in a way that hasn't been able to be done at scale before. What about the future? Well, we've been asked to do more samples, and there's more to be learned, especially with bivalent vaccines coming through. And we could do that. But there are other platforms after 2 years that can also do that. And the amount of money pouring in is enormous. And the skills and expertise have really got focused onto COVID. And to be honest, I think what we're doing is going to take a back seat. So I've sort of handed over this enormous collection of samples and precisely phenotyped subjects to the immunologists and the microbiologists and the infectious disease doctors rather. I don't think there's going to ever be a cardiovascular impact again, but as people and scientists, we've been changed by this process, and this sort of ask the big questions, assemble the teams, goal-focused deadlines, trying to bring that into our day-to-day work. And the one area I'm doing now is artificial intelligence for medical imaging. We're all using AI every time you use your phone. Those targeted ads are reading the photos you post. And if you post a photo of a dog, you'll get an advert for dog food, you know. But in healthcare, the AI has to be explicable, it has to work anywhere, it has to be usable. We have to trust it. And you have to get it so that you can make it available. And it's got to be in a worthwhile place where it makes a difference, like choosing who has treatment. And so using the lessons from COVID, funded by obviously the Guy Foundation and the pharmaceutical company itself, those sort of lessons have been translated into how we're doing things. And sitting in an academic environment on our own, thinking of things and doing little sample studies and maybe networking with a few people in the US is not enough. And we've seen the power now of industry, and we've done a spin out, and we're trying to really start to try and be goal orientated for making a difference in society in a way that we wouldn't have felt brave enough to have done before COVID and the lessons learned from this project. So thank you very much. This was the world's sort of most granular longitudinal study of mild disease, and by the metrics of the publications, I mean, I've shown you there are actually 3 Lancet, 3 Science, and a Nature, for example. It was the most successful study of mild disease. And I've shown you an emergence strategy. I've alluded it, if you don't mind, as being a hell of a good fun thing to do, but we were driven like there was, you know, the devil biting our tail as we were running along. And you saw how we funded it and the adaptation we did and the survival as we moved along. And so thank you for making it happen. And I hope you enjoyed the story. And now I'm back to cardiac MRI. So keep calm and get involved in research. Thank you.
James, thank you very much indeed. And as you say, back to your day job, quite amazing, that program. We were delighted that you approached us, I think day 2 or something, for the funding. And I think we took all of half an hour to agree, I think it was.
You're great.
To do it, so we were delighted to do that. Nina has just, and we will take questions for your paper at the end. Nina has just reminded me that, having said I'd introduce both of our speakers together, I forgot to introduce Alistair. So Alistair, if you'd like to just prepare your get your slides up, I'll speak over that whilst you're putting your slides up. So as many of you know, Professor Alistair Nunn is our Director of Science for the Guy Foundation. He's also a visiting professor in quantum biology and bioenergetics at the Research Centre for Optimal Health at the University of Westminster, and works with colleagues Jimmy, Reese, Iffy there. Alistair is a theoretical biologist who loves nothing more than asking the big questions about life and the universe. So in today's talk, he's going to set out the scene for the series by talking about the quantum mitochondria. So Alistair, over to you.
Okay, can you all see the screen? Have I got the right screen, Nina?
Yes, you have.
Yeah, I'm good. Okay. Well, okay, James, I'm not quite sure I can follow that. I certainly quite haven't got the altometric metrics that you've got. Okay, so over to the theory. As Jeffrey suggested, we very much have been into mitochondria and what they do, and certainly in more recent times we were very keen on looking into the quantum biology and everything, and certainly with COVID that gave us a real time to start thinking about other things in terms of medicine and how potentially viruses might work. So in this little talk I'm going to put forward a few ideas, and I do apologize, some of them may or may not turn out to be right, but I think they might be quite thought-provoking, so I hope that people understand where we're coming from. So let's start. Long COVID, what is it? I'm sure James can do a much better job than I can. But in essence, it's you probably all heard about it. It seems to be a post-acute sequelae of a viral infection, and statistics vary depending on definitions. But it seems that over and beyond 6 months, something like 10 to 15% of people develop symptoms which are well after their acute infection, and these include certainly things like exercise intolerance, tiredness, brain fog, which I get all the time anyway. I don't need a virus to have brain fog, loss of smell, and certainly biochemically there are redox imbalances. And one of the underlying commonalities is obviously a chronic inflammatory condition and clotting imbalances. And so one of the questions is, of course, what's happening and what's the central connection to all of this? So, of course, we're very much into mitochondria, and soon as this outbreak broke and James and Jeffrey started talking, we started thinking, well, what about COVID-19 and mitochondria? And we put together a little paper which built on our previous experiences and interest, certainly around lifestyle-induced disease, because one of the things that became quite apparent is that severity of the COVID-19 seemed to be related to metabolic syndrome and incidence of obesity, and certainly comorbid conditions. Of course, this immediately made us think about mitochondria. And certainly primary mitochondrial dysfunction, certainly conditions where you've got mutations in both mitochondrial and nuclear DNA encoding genes involved in mitochondrial function, can predispose to more severe disease. But age is one of the big ones, and certainly all evidence in terms of aging is that mitochondrial function does degrade with age, unfortunately. But the one we were quite interested in, of course, was the environment-induced one. And this is, of course, the idea that a poor lifestyle might induce something called the metabolic syndrome, which is associated with mitochondrial dysfunction. And we had a look at this because we've also been quite interested in cancer. And one of the things that struck us, certainly from the early publications, was there seemed to be a link between a shift from oxidative phosphorylation towards more aerobic glycolysis, which could favour replication of the virus. And so we mentioned this in our paper. It indicated some kind of metabolic tipping point, which could centre on mitochondrial flexibility. So this got us thinking a bit further. And as many of you who did biochemistry, so I agree that I understand many of you obviously haven't done biochemistry, but one of the things you often see in textbooks is this picture here of a virus infecting a cell, and somehow it goes in and it half inches all the metabolism of the cell to produce more viruses. And one of the things you don't often see in many of these textbooks is the role of the mitochondria. But of course, for a virus to replicate, it's got to completely manipulate everything in the cell in favour of itself. And one of the really interesting things is that, and this follows on from Nick Lane's work, and Nick Lane's presented some of his work before with us, and this is this idea that the Krebs cycle, which many of you may or may not have heard of, is a very ancient biochemical cycle in the mitochondrion, which in many textbooks, original texts, which are saying, oh, it's very important in generating energy, and it's electrons generating energy, which creates the proton gradient, and that's all very good, and it creates ATP. But what people don't realize, of course, is also absolutely essential in the way it rotates in generating precursors for metabolism and biosynthesis of new compounds. And Nick Lane has actually written this new book called Transformer, and I have to, hats off to him, because he's focused it on the origins of the Krebs cycle. And immediately you start to think, okay, well, it can run in two directions. And the point he makes is quite likely that the origins of life, it was running in a complete different direction, which was biosynthetically. So what we've got in the textbooks is actually opposite way around to what actually started the origins of life. But critically, it links in very well with the commonality which many biologists know about, which is between the growth of cells and inflammation, and they share many of the same biochemical pathways. And one of the things that initiates these pathways is hypoxia. And of course, this means follow the electron. Of course, all of you familiar with quantum biology will think, ah, electrons, here we go. So I just thought I'd just quickly cover the origins of life. And this is, okay, there are other theories out there. Other theories are available if you care to look for them. But we're going to follow the one which is based on alkaline thermal vents. These are deep sea vents which are thought to have been around at the origins of well, shortly after the Earth cooled, but certainly is still available now. But the chemistry on the planet was very, very different. And one of the key things about these thermal vents was that they had a source of hydrogen coming up from the Earth's interior, meeting a very carbon dioxide rich sea. And this is a very big potential, energy potential. And when you combine this with theories of self-organization and path equilibrium dissipative structures, and that's a picture of Ilya Prigogine, who's one of the originators of these ideas and the maths behind it, you can start to see that life is actually a dissipating structure which follows the basic laws of thermodynamics. And if you put all of this together, you can start to see that actually a lot of the chemistry we see in life could have happened long before there was proper life as we define it. But over time, this did lead to life and bacteria and archaea, and eventually it also led to the formation of photosynthesis. Now, again, there's a big discussion going on in the world of origins of photosynthesis. When did it occur? Where did it occur? What is interesting about this is, of course, that thermal vents, and this is again, other sources of photons are available, but we'll say thermal vents actually glow. They produce a lot of light. You might not be able to see it, but sensitive cameras pick this thermal light up. And this actually changes things because there's quite a good theory out now about actually photosynthesis started in the deep sea, which is not anything everybody realizes. But anyway, the offshoot of this was these early bugs and bacteria could split various other chemicals to get electrons because they need electrons to grow. But in time, they started to split water. And as soon as they started splitting water, and I think Steve Thorne will obviously back me up on this one, they started to produce oxygen. And this changed everything. At the same time, of course, all of this was going on. And this is the same theories. If we go from thermodynamics, we can also look at quantum mechanics. And so, well, actually, was it important in the origins of life? Well, there's evidence that some of the very early chemicals involving iron and iron sulfides have some very interesting quantum structures. And these are absolutely essential in modern life. And one of the interesting properties of these compounds, they could well enable tunneling and quantum tunneling, which has led to the whole idea that certainly in your electron transport chain in mitochondria, there could well be quantum processes like tunneling going on. But of course, there are other things which involve quantum chemistry. And this is, of course, how the very first precursors to the Krebs cycle started on mineral surfaces. And the mineral surfaces require, has some quite unique properties which enable quantum mechanics to generate early chemicals. And of course, then you start to invoke things like, well, if there's tunneling going on, is there, are electric fields important? What about spin and everything like these other very strange quantum properties, which Bethany can explain a lot better than I can. Anyway, this all led to a system which seems to have some basis in quantum mechanics, and certainly it can be described by thermodynamics. And of course, one of the very earliest interests in quantum biology was in photosynthesis. So all of this is encompassed here. Anyway, let's move on a few billion years. And again, timings are fairly vague here, but it's about 2 million years ago. Two kingdoms got together. One was the bacteria, and the other was the archaea. And these got together to form the eukaryotic cell. And this is the nucleated cell, which we all know and love today. And I think about this, of course, in these cells, the very basis of metabolism is something similar to the Krebs cycle. And of course, it can run in, and it seems to run in two directions. And one direction is biosynthetic, the other is to generate energy. And of course, another really interesting point about all of this is, and it's something that Nick Lane points out, is of course, if you think about it, you've got a cycle. Can it run in both directions at the same time? And this is a problem which is sort of worried people for a long time. Well, the answer is it's actually probably more complex than that. But one solution is to have different cells doing different things. And a eukaryotic cell in a multicellular organism, of course, can do biosynthetic in one organ, or it can be producing energy in another. Or indeed, even within a cell, it can be mitochondria could be doing both. So it gives you a lot of flexibility. Then we move on again, and along comes our spiky nemesis. And I just put Darwin and Wallace here. They would have understood the evolution behind this. I'm not going to go into too much detailing. But one of the key things, of course, is that the virus itself needs to induce metabolic reprogramming, and which, of course, then means it needs to trigger off a system which enables it to stop causing its host cell to generate things which enable it to grow. And one of the fundamental principles is hypoxia. If you have a stress in a cell, it can start to use electrons in a different, or shove electrons in a different direction. And the cell or the system within it can detect that as hypoxia. And hypoxia is just a lack of oxygen. It runs out of an electron acceptor. So this is a very old system. Now, certainly in us, and if you get initial infection, if it goes into the respiratory system, it's a respiratory virus, we can activate our immune system, and that can go, for instance, in what they would call the reverse direction, start generating more viruses, which is fine in say that cells are infected in your immune system. But if your whole system then tips over into what would be a chronic inflammatory system, the various signals can start to induce this program in different cell types. And some of these cell types, in particular in the brain, the muscle, the heart potentially, and even platelets, can start going into what will be the biosynthetic pathway, which of course might debilitate their energy production. So this could be quite serious. And if we look at this from, again, some old biochemistry, and this chap here is Otto Warburg. Now, if you remember, I briefly mentioned this idea of aerobic glycolysis, and of course, Otto Warburg was very famous in saying, well, actually, cancer is a disease where there's mitochondrial dysfunction, and for some reason, the cell, rather than using respiration, starts using glycolysis. He was slightly wrong on some of it, but his point was well made. And it turns out, I think Otto Warburg knew Hans Krebs, who was one of the originators of this cycle. So it could be that, and we're not alone in this thinking now. There's several other groups thinking, well, actually, this could be quite important in this switch to metabolic programming. And certainly for many viruses, it seems that the go-to standard of practice is to start metabolically reprogramming your host. And although this could be seen to be critical, say, in inflammation, it's also what cancer uses. So in a way, you start to see the viruses as having some commonalities, inducing a kind of metabolic disease, as cancer may be viewed as. And I know cancer has mutations, but you could argue that it's also a metabolic disease. And if you go back to hypoxia again, that's how life started. So what you're seeing, there's some kind of crossover with viruses reverting the system to something which is very primordial, which is again an old idea which has been around for cancer for a long time. Simply, cancer is a reversion back to a very old piece of metabolism. And if we then go back to our mitochondria, this is one of the things that we've for many years trying to push forward, is this idea that mitochondria aren't just batteries producing energy. They're actually central to everything that goes on the cell. So they're involved in immunity, inflammation, they control cell death. They're involved in producing all the essential precursors to proteins, to lipids, to nucleotides, and they also control iron flow. For instance, mitochondria key in calcium signaling, and any biologist will tell you calcium is really important in signaling. They're involved in that too. And so when you start stripping away all the complexity, you come back to something quite simple, which is this rotational cycle. And in a way, mitochondria do actually reflect the very early Earth, if you think about it, and stand back and go, well, actually, yeah, they're like a battery in one sense, but it's a battery, it works both ways. You can rebuild the battery by reversing it. But of course, one of the things that's really key here, and this is something which, again, is only really being discovered, although I think Douglas Wallace and Wallace has been talking about for a long, long time, is of course, is the epigenetic thing, is that all the components in the Krebs cycle can filter out and go to the nucleus and reprogram your nucleus. So there's this lovely tight cycle between what happens here and what happens in the nucleus. So just to quickly go through, just to bring this into context, which is important in looking at what we might think going on, is of course, when we start to think about aging, and of course you can argue that many diseases seem to accelerate the aging process. And thermodynamically, this is simply a process whereby if you've got a network, one of the network nodes starts to fail and the whole system recalibrates. And of course, if we then go back to the quantum world, you can argue, well, what is aging? Aging is a loss of structure. And this is something which is one of the theories that people put forward into aging, is that, of course, with time, we lose the ability to maintain our structures and say proteins. And one of the key ones, for instance, in the mitochondria, is that over time, one of the early complex ones doesn't work quite as well. And we try and regenerate it, but with time, that process is not doesn't have high fidelity, so it eventually fails. And if we think about the quantum underground, i.e. the ability, for instance, to tunnel electrons, that system slowly degrades. And of course, it's the movement of electrons again. And it suggests that the fitness of something is determined to some degree how you control your electrons. And of course, if that deteriorates, you get to a point where the system can't cope anymore and it fails. And of course, this then comes subject to natural selection. And critically out of all of this is, of course, that this is almost scale invariant. If you just go back to the maths and thermodynamics and forget about biology, you just look at nodes. Every component from your protein up to your, say, your mitochondria, to your cell, to your organs, to you, to your species, even to life, is disposable at some level if it fails. And the dinosaurs knew about this very, they realized this quite quickly. But humans are quite dispensable. And if it gets damaged, we revert to simpler things. It's always been said that, if we have a nuclear war, what's going to be left? Cockroaches and bacteria. They'll revolve. So it's quite sobering, really. Now, there's another concept here, which I think we've talked about before, and we've always loved this idea. And this is this concept of hormesis, which is effectively a biological adaptive process to stress. What doesn't kill you makes you stronger. And again, from evolutionary perspective, it makes perfect sense because you have to adapt to a changing environment. But what's quite interesting, of course, it's almost the flip side of inflammation, because inflammation is basically a system to restore homeostasis. If something's damaged, it has to be rebuilt and regenerated. And so these two concepts kind of go together. And it also goes back to this idea that thermodynamics, if you've got something which is not functioning, it can result to a simpler system to regenerate. But also what falls out of this is this idea that, and this is due to something called adaptive thermodynamic dissipation. A node, if it's damaged and doesn't work, it seems that through the maths, that this system will actually be removed, or in fact, it will remove itself. And it turns out that this idea of self-destruction, what we might call suicide, is actually something which has been around a very, very long time. So aging is linked into the removal of particular components. So what's this all to do with Krebs and everything? Well, it can be argued that hormesis is a system which is triggered by a change in electron flow. And obviously, because Krebs cycle is all about moving electrons around and energy, that system must be intimately involved with it. And it's a back to origins idea again. If the system gets damaged, it goes back to its simplest system, which in our case may well be reverting back to a more primordial system. So sorry about that. So hopefully that gives some idea of what's going on. So what can we say long COVID is? And again, this is a hypothesis. It's an idea. I was talking to Jeffrey about this the other day. It could be viewed as a partly non-resolving epigenetic state that reflects the hypoxic beginnings of life. Resolution may well be more difficult if the starting system is not very fit and is already compromised in its ability to flow electrons to oxygen. And of course, I'm referring back to, for instance, if you've got the metabolic syndrome, you're not quite as metabolically flexible as you could be. And interestingly, it could reflect a very ancient mid-state, which initially, in terms of us, initially favoured the paradoxical viral replication resolution, but gets stuck. So what I'm saying here is that a virus, if it wants to replicate, needs to induce a slightly more ancient paradigm of metabolism. But equally, the same system is the same system which recognises there's damage, which is inflammation. And if we go right back in time, it seems that there was a stage in, say, Precambrian life when this condition existed, and there was, in the primordial sludge, there were organisms which survived because they could balance these two things very well. So this, this again is an idea that reflects a very ancient epigenetic state, for instance, of life. So that's just pure speculation. But what does it mean in terms of resolution? Well, one of the key questions here is how do we, if you've got something like long COVID and you're having these symptoms for years, for a long time, how do you get the switch back to where it should be? And one of the suggestions could well be here that you need to induce an adaptation. And this, of course, falls in with hormesis. And if we think about the Krebs cycle, one of the key things that certain things like exercise and calorie restriction do is start to force the Krebs cycle back in the direction of energy reduction. And interestingly enough, there are many plant chemicals which kind of mimetic this. They're mimetics. They do it a little bit because they're stress compounds in the plant. And which tends to suggest to us that if you want to start getting some kind of resolution, exercise is going to be key. Now, this is not a new idea. The literature is now full of it. There are opposing theories saying you shouldn't do it at all. I think like a lot of these things, it's a question of dose and getting the timing right. Calorie restriction could be quite powerful in the sense that if you stop loading the systems up with calories, it can't build anything as such. Therefore, it has to start switching back to a model where the Krebs cycle, for instance, goes in the forward direction. And certainly, it also stimulates the system to regenerate its proteins. One of the key things we know in aging is that there's this process called autophagy. And this is where components in your cell start to be regenerated. They start to be taken apart to use and regenerate for new bits. And basically, it's like autophagy means auto-eating. Mitochondrial turnover is a very important component to this. So all of these processes stimulate this process, which could be very much linked to the Krebs cycle. And certainly, if this is the case, one of the key components of this is it might actually then enable different organ systems to go back to where they're supposed to be. Now, if we're saying that generalized inflammation induces metabolic reprogramming in all the wrong organs, this process might stimulate that process back in the right direction in different organs. So we might be able to, A, get our immune system rebalanced, B, get our exercise tolerance back, and certainly, for instance, get our inflammatory coagulation system back in balance. There are also a few further thoughts that come out of this. We kind of touched on it briefly before. Have viruses evolved to induce very, very mild hypoxia? Because actually that will push the Krebs cycle in a direction which enables their growth, which of course has a knock-on effect. And again, there's some evidence for this. One of the problems, as James alluded to, of course, is that, and certainly we see this both in acute COVID, but in long-term COVID, there seems to be this slight imbalance in coagulation. And not many people know this, or everybody knows this. Platelets contain mitochondria, and they show a metabolic shift when they become activated, which suggests that, well, if your mitochondria aren't healthy throughout the body, they might not be healthy in your platelets either. But what we do know, and this is a new area of research, is that mitochondria are actually being swapped between different cells all the time. And they don't just sit in cells and do nothing. If a cell's damaged, it can actually ask another cell to give it some new mitochondria. And not only that, the components of these mitochondria and the things that control them, like short transcripts of RNA, can be moved around in things called extracellular vesicles. So in fact, there's a much more dynamic structure going on here within the body. And so if you, for instance, take your exercise, you stimulate certain ones in your muscles, and they can actually then stimulate this process right throughout the body. Now, one of the interesting things about the exercise, of course, it signals to the rest of the body, probably in some of these mechanisms, but also through well-described cytokines and myokines, that it needs to reset the metabolic programming. There's some quite new research, for instance, on something called irisin. And irisin is a peptide which is released by exercise, and it has a lot of quite good healthy properties. One of which, and it's been shown quite recently, it can slow down or reverse Parkinson's. So of course, there's all sorts of things of looking at the myokinome, as it's called, from exercise, which might be something we look at. But of course, the opposite side of this is that, of course, the virus could be accelerating the aging process. And this is something called inflammaging, which is a well-described process. And it seems that as we age, we become more inflamed, and it sets up a kind of vicious cycle. So you need to perhaps slow that process down. And a slightly more, I suppose, philosophical point here is that if we're right about the viral thing, viruses have been around perhaps since the year dot, and they could be seen as entities which actually accelerate the dissipation process, and not only accelerate aging in us, but potentially the heat death of the universe. Now, people might think this is going into the realms of science fiction, but if you believe that life is actually accelerating use of energy, that is not an entirely implausible theory. And so finally, really, the take-home message, really, and this again, with thanks to Nick Lane, is he talks about in his new book something called the flux capacitor. Now, most of you are probably familiar with this from Back to the Future, but his point here is, if you look at a mitochondria, that's exactly what it does. There is a flux of metabolites through it, which extracts energy, which can be used for both producing energy for movement, but also producing biosynthesis of new compounds. And a key outcome of this is a very high potential across the membrane, acts as a capacitor. And that capacitor tells us a great deal and tells the body what it needs to be doing. If it drops off, it needs to do one thing. If it gets too high, it needs to do something else. And certainly another message that comes out of this, if we start to think about mitochondria as organelles which both work as energy producers, but also in the biosynthetic pathways, they will always be slightly compromised. And if their total capacity is decreased, then of course, what are they going to do? And we certainly know that if you trigger their development, trigger their renewal, you increase the size of what we would call the mitochondrial metabolic engine, and it has more flexibility. And it's a bit like taking a small car like this Fiat 500 and driving it round and round a track, parking it in the garage every night. And next morning, it's turned into an Audi. The day after that, it's turned into Ferrari. And 3 weeks later, it's turned into a Formula One car, because it increases its capacity to do work. And certainly this is the idea, this is taken from another paper. But if you're in a situation where you've got very few functional mitochondria, they get very heavily overloaded. They're just like an electric bus. But if you start to stimulate having more of them, they have more capacity. And in a sense, this is what may increase the size of your hormetic engine. And this may well be your metabolic engine. This may well be something which we need to think about when we look into for things like long COVID. I'd just like to say thank you very much. That's the end. Thank you very much to the University of Westminster and Jimmy Bell for supporting, Guy Foundation and Jeffrey for enabling me to have these kind of weird thoughts. And I would also like special thanks to Nick Lane. And he has really, over the years, through his very many very good books, made us think completely outside the box. So anyway, there we go. Thank you very much.